Now, the whole rest of this unit and this note packet is about osmosis, the movement of water, and how the movement of water can affect cells. And the movement of water is so important, and it has such an effect on our cells that we dedicate this whole last part of this unit to the movement of water, to osmosis. Now, I'm going to go through some rules to help you predict which way is water going to move. And you already learned the first rule is that water always moves from an area of high concentration of water to an area where water is in low concentration. And you've seen this diagram before. There's a high concentration of water here. There's a low concentration of water here. So there's going to be a net movement of water in this direction. Now, sometimes I just show you water molecules like this, and you can see that there's definitely more on this side than on this side. But sometimes we just give you water concentrations, and we say this side, or solution A, is 90% water. Solution B is 50% water. And you have to just remember this rule that water moves from high concentrations to low concentrations of water. So in this situation, water is going to have a net flow of water in this direction. Now, the reason why I have to teach you other rules is because sometimes we do not give you the percent of water. If we don't give you the percent of water, you're going to have to apply some other rules. So here's rule number 2. Sometimes we say that a cell or a solution is hypertonic or hypotonic. So then you have to have this next rule memorized. You have to remember that water is always going to move from a hypotonic solution to a hypertonic solution. Now, to help you memorize that, and trust me, someday when you're sitting in your college biology class and they're talking about hypertonic and hypotonic solutions, you're going to remember this rule. So imagine if you were going to go to a party this weekend and you have 2 choices between which party you want to go to. One party, you text people and they say it's kind of boring. You text someone else at a different party and they say it's a great time, everyone's having fun, everyone is hyper. You're going to go to the hyper party. So remember that water is going to go to the hypertonic solution. So let's just write over here to help you remember this rule. You want to go to the hyper party. Now, there's really no such term or no such thing as a hyper party when we talk about biology and in science. So don't write that water goes to the hyper party. You need to remember water goes to the hypertonic solution. We're just going to use this hyper party to help you remember this rule. Now, when I'm talking about hypertonic and hypotonic solutions, then I'm talking about how much water there is compared to solutes. So let's review real quick. If we have dissolved molecules in a solution, they're called the solutes. And remember, water is sometimes called that universal solvent because it dissolves so many different molecules. It's called the solvent. Now, a hypertonic solution is a solution that has a higher concentration of solutes compared to another. So it has more solutes dissolved in it compared to another. So imagine if you have tea and then you have a glass of tea that has sugar in it. The one with the sugar in it is going to have more solutes in it. It would be the one that is hypertonic. Now, to help you remember that, I like to think of that old wives' tale, that if you give kids a bunch of sugars, then they're going to be more hyper. So think of more solutes, like sugar. That's going to be the hyper one. So by default, the solution that has a lower concentration of solutes is going to be the hypotonic solution. And anytime we have 2 solutions that have the same concentration of solutes, then we say those 2 solutions are isotonic. You probably know what that prefix means because in geometry you probably learned what an isosceles triangle is. And that was a triangle that has 2 equal sides. So iso when we see that prefix iso, we're talking about something that's going to be equal, or two things that are going to be equal. So these are examples of problems that we can ask you, and we're going to ask you to predict which way water is going to move. So we made a solution, and that solution was hypotonic, and we placed it in a dialysis bag. And a dialysis bag is kind of like plastic Ziploc bag, and it's got small little holes in it. So if the inside is hypo, then the solution that we place on the outside by default is hyper. Now you have to remember, water is going to go to the hypertonic solution, just like you're going to go to the party where everyone's hyper. So we're going to show water's moving out into that hypertonic solution. So let's label that arrow with H2O so that we know we're talking about water moving, because we're talking about osmosis. So if I give you a situation, I say a cell, so here's a cell, is surrounded by a hypotonic solution. Predict the movement of water. In this situation, water is going to move into the cell because the inside is that hypertonic solution. Water always goes to the hypertonic solution. Now, just because a solution is labeled hypertonic, it doesn't mean that it's always going to be hypertonic. So, for example, if I have just plain water compared to a 3% sugar solution, obviously water is going to be the hypotonic solution, and sugar is going to be, in comparison, the hypertonic solution. Now, if I take that exact same 3% sugar solution, if I compare it to a 10% sugar solution, then the 3% sugar solution now has less solutes in it. It is the hypotonic solution, because that 10% sugar is the hypertonic solution. So when we're talking about hypertonic and hypotonic, those are terms that we use to compare different substances. So just because you label a solution hypertonic doesn't mean it's always going to be hypertonic. It's just like you might say I'm short, but if you compare me against someone that I'm taller than, then I'm no longer short, I'm tall. So that was rule number 2, and now we're on to rule number 3. So take a look at this diagram that I've given you right here, and you can see that I do not give you water percents. I'm not showing you that's like there's 90% water or 50% water, and I'm not showing you there's a hypertonic and a hypotonic solution. I'm giving you solute amounts. If I give you a problem and I ask you to predict which way water is going to move, and I just give you solutes, you have to remember this rule that solutes suck water. Wherever there's more solutes, then that's the way that water is going to move. Water is going to move to where there's more solutes. So think solutes suck water. So here's a U-tube, and I have a membrane that's separating solution A and solution B. If you add up the amount of solutes over here, it's 0.9 molar. Over here, we have 1.2 moles of solute. So there's more solutes on this side. Solutes suck water. Water is going to have a net direction or net flow from side A to side B. Now, some of you maybe picked up, well, I already know that if there's more solutes, this side is the hyper one, and this one's the hypo. And if you thought about that, that's great. So if you know that this side is hypertonic because there's more solutes, water is going to go to that hypertonic solution. So look at some other diagrams here. So it looks like we have all water over here, and we see we have some solutes over here. So this is an easy one. Since there's more solutes over here, solutes suck water. Water is going to move in that direction. You also maybe determine this side is hypertonic compared to this side. And water goes to the hypertonic solution. A few more. So let's predict which way, again, water is going to move. There's more solutes inside. Solutes suck water. So let's show water's moving into that cell. And we're going to do some math here. We have 45 grams of solutes inside, 30 solutes outside. So there's more solutes inside. Show water moving in. That inside is hypertonic compared to the outside. Now on this bottom diagram down here, you can tell that it looks like the concentration of solutes is the same both inside and out. So if some water moves in, some water is going to move out at the same rate because these two solutions are isotonic. Remember, solutions at the same concentration are considered isotonic solutions. And then one more diagram. It says we have pure water on the outside. That means it's 0% solutes. Obviously, we have solutes inside. This would be hypertonic. This would be hypotonic. Solutes suck water. Water is going to move into that cell. Now that we've talked about hypertonic, hypotonic, and isotonic solutions, we're going to talk about how those solutions can affect cells. Now, if you remember, hypertonic solutions compared to hypotonic solutions were solutions that have a higher concentration of solutes in them compared to that hypotonic solution. So it's really important that organisms are regulating exactly how much water they have in their system and how many solutes or what their concentration of solutes are in their body, because if we get situations or environments where there's too many solutes, that can pull water out of your cells, and we really don't want a net flow of water moving into the cells or out of the cells. So if we take your cells, animal cells, and we place them in a hypotonic solution, the reason why I wrote hypotonic with this big capitalized O is because I want to make sure you understand that if you take a cell and put it in a hypotonic solution, this is what's going to happen to the cell. It's going to expand. So let's look at this diagram down here. It's telling you that we have cells and we've placed them in a hypotonic solution. So if the outside is hypotonic, then that means by default the inside is going to have more solutes in it, and it's going to be the hypertonic solution. If you remember, water goes to the hypertonic solution. Remember what party you want to go to. You want to go to the hyper party. So in a hypotonic solution, water moves into animal cells, and animal cells can become so full of water that they break and they can die. So hypotonic solutions make cells do this. Now, a hypertonic solution is going to do the opposite. A hypertonic solution is going to make cells shrink. So that is why I made my E look like this. I made it small. I want you to think of it as being an O that's shriveled up to look more like an E now. If I take an animal cell or plant cell and I put it in a hypertonic solution, so that means that the outside of the cell is now hypertonic, the inside is hypotonic. Water always moves to the hypertonic solution from a hypotonic solution. Water moves out of these cells, and these cells will shrink up. For us, for animals, anytime that we put a solution into your body, we always want to make sure that it's isotonic compared to your cells. So now if your cells are surrounded by a solution that's isotonic compared to your cells, that means the outside solution and the inside solution are isotonic to one another, and there's going to be an even movement of water. If some water moves into the cells, then some water is going to move out, and those cells are going to be able to maintain this normal shape. Whenever animal cells break, like you see here, we actually have a term for that. It's called cytolysis or cytolysis. Cyto means cell. Lysis means to break. So you can probably figure out why we term this cytolysis. Cyto, cell, lysis means to break. Whenever our cells shrivel up, then the term for that is called crenation. Now, to help you remember that, I think this word, this word reminds me of cremation. And if you think about it, if someone is cremated, then they're going to end up becoming much smaller than what they were whenever they were alive, because we're going to reduce them basically to ashes, and they're going to be able to fit into an urn on someone's shelf. So we make you smaller when we cremate you. So crenation is whenever these animal cells get smaller, whenever they shrink up. Now let's talk about plant cells, because they have a cell wall, then they're going to react a little bit differently whenever they're placed in these different types of solutions. Again, if I take a plant cell and I put it in a hypotonic solution, so if I pour water on it, hypotonic solution, by default that means the inside is hyper. Water moves to that hypertonic solution and plant cells are going to swell. If I take a plant cell and I put a hypertonic solution on it, water comes out because the cell is hypotonic, the outside's hyper, and water moves out to that hypertonic solution, and these plant cells shrink. Now in this situation, whenever the plant cell is swelling up with water, that plant cell can't rupture because they have that added accessory there. They have that cell wall, and that prevents their plasma membrane from getting too big to the point where it can actually rupture. And this is actually the best situation for plant cells. For animal cells, we need to surround those cells with isotonic solutions. But for a plant cell or plants actually prefer hypotonic solutions. And here's the reason why. Is they're built of cells and they don't have any kind of skeletal structure to give them shape. We have a skeleton to aid in our structure, but they don't have that. So they literally are just built of cells. And so these cells have to be very, very tight so that these plant cells can form their structures and they can be upright. I tell students to think of if I asked you to build a tower, and I asked you to build a tower out of marshmallows, or if I asked you to build a tower out of wooden blocks, and you had a race to get this done before another group, I think most groups would choose to build out of those wood blocks because they're sturdy, they're hard, and you're going to be able to stack them a lot easier and make this upright structure. This is kind of like, this cell right here would be like a squishy marshmallow. Imagine trying to stack a bunch of squishy marshmallows. Like, it's just going to fall over. So plants actually prefer a hypotonic environment so that their cells become nice and tight and full of water. So here's a situation where we have an isotonic compared to the plant cell. So if some water moves in, some's going to move out, but this cell is actually going to be a little more squishy than this one over here. Now remember the animal cells, we had terms for when the cells were breaking or shrinking. So it's cytolysis, when the cells were breaking and dying. It's crenation, whenever they're shrinking up. Plant cells also shrink, but we have a different name for it. We call this plasmolysis. And it's more named after the fact that the plasma membrane shrinks away from the cell wall whenever the water is leaving the cell. It's kind of named after the plasma membrane, the fact that we have kind of the shrinking of the plasma membrane. Another difference, and another reason why we term these with two different terms, is because an animal cell, if it's suffering from crenation, it dies. It doesn't really come back from that. But if a plant cell is suffering from plasmolysis, if we add water to it, then that cell membrane will move back up against that cell wall as that cell fills up, and the plant cell can recover from that. It usually doesn't die whenever that plant cell has been plasmolyzed. So let's just review those terms in the next blanks. The animal cells bursting and dying, that was cytolysis, so cell lysis, or cell breaking. Animal cells shrinking, think of cremation, you'll shrink up, but it's called crenation. When the plant cells shrink, it's because their plasma membrane is pulled away from the wall. It's called plasmolysis. Now the last topic that we're going to talk about is Turgor pressure. And when we talk about Turgor pressure, we can only talk about plant cells. Plant cells have the ability to experience what we call Turgor pressure, even the increasing in Turgor pressure or the lowering of Turgor pressure, because the definition of Turgor pressure is pressure exerted on the cell wall by water in the cell. And animal cells do not have a cell wall, so technically they can never develop Turgor pressure. So Turgor pressure is really just related to plant cells. If a plant cell has high Turgor pressure, like this one does right here, then you have a very full, a very tight cell. And this is actually, again, normal, or what is best for plants. If you have a plant cell and it has low Turgor pressure, then there's no water inside putting pressure on the cell wall. I'm going to draw that on this one. There's water inside, so there's pressure put on that cell wall. So this is what we mean by Turgor pressure. But since the water is coming out of this cell over here, then we don't have this buildup of pressure due to water inside the cell. And so this is a squishy cell. It's really limp. It's really flaccid. This is actually what the cells are going to look like of a plant that's in your house that is wilted. So a wilted plant has plant cells that have low Turgor pressure. And so these cells are really squishy, and that plant's leaves will drop and it'll fall over because it's not, it doesn't have those tight cells to keep it upright. So again, let's write high. I'm going to put TP for Turgor pressure in this cell. This cell is experiencing low Turgor pressure. You can also relate this to something like water balloons. If you put a lot of water in a water balloon, then there's going to be a lot of pressure in that balloon. It has high pressure, high Turgor pressure. If you take water out of a water balloon, then it's going to be really squishy and it's going to have really low pressure or low Turgor pressure.